Afternoon, I'm Míriam Barrabés, and I'm going to present you my work on machine learning for genomic sequence processing, which has been co-advised by Xavier Giró i Nieto from Universitat Politècnica de Catalunya and Daniel Mas Montserrat and Alexander Ioannidis from Stanford University. First of all, I will start by defining what is the genome, since probably most of you are very familiar with machine learning, but not so much with genomics. So the genome is the set of instructions that forms an organism. An organism is made of cells which contain the nucleus, and the nucleus contains the chromosomes. And the chromosomes are made of DNA material. DNA has two strands that form an helix, and DNA has a sequence of base pairs, adenine, which always pairs with thymine, and cytosine, which always pairs with guanine. So since these base pairs are complementary, actually we don't need the information in both strands, but only in one of them. And so DNA can be represented as a string, as a sequence of characters of A's, T's, C's, and G's. And DNA has many positions in the order of billions, but we are not interested in all of them because most of them do not contain variations between individuals of the same species. So we are only interested in around 2% of the genome, which contains variations. And these positions, they are called single nucleotide polymorphisms, also referred to as SNPs. And in each of these positions, there are two bases that are possible. The base which is common in the majority of the population, it is called the reference, and it is encoded with a 0. And the reference and the base which is less common in the population, it is called the alternate, and usually it is represented with a 1. So the genome is very related to our health. For instance, it codifies our risk to have a certain disease, for example, hypertension, also our response to medication, and even the risk we have of dying from COVID-19. So genomic studies applied to health have application in precision medicine, which is kind of the future medicine and consists in providing a personalized prevention and treatment of a disease based on the characteristics of an individual, including the individual genome. And the genome also codifies for our phenotypes, which are the unique characteristics that result from an organism genotypes. The genotypes are sets of genes, and examples of phenotypes could be the color of our eyes, the sweetness of a fruit in a plant, or the percentage of fat in an animal. So studying the phenotypes has applications in selective breeding, which consists in selecting the desired traits during the breeding process between the males and the females. And this has a lot of applications in plants, but also in animals. And in fact, it is usually more convenient to work with plants and animals data more than humans, because they have less privacy restrictions, and also it is less controversial. And the same techniques can later be applied to humans. So next, I will talk about my first work, which consists in predicting dog phenotypes from their genotypes. So given the genome of a dog, we aim at predicting its breed, its height, and its weight. And this has been studied before, but there are some questions that haven't been answered, which are hundreds of thousands of input features necessary to predict breeds and phenotypes? This question is relevant because the price of genomic sequencing is proportional to the amount of positions that are sequenced. So basically, knowing how many positions we need helps reducing the cost of our study. And the second question is, do nonlinear methods outperform linear ones? So this question is relevant because it helps you understand more the distribution of the data and the relationship with the labels you want to predict. So the data for this study consists in around 400 samples. Each sample is a dog DNA sequence with around 200 SNPs. And here you can have a look at the data with a PCA, and TSNE. And next I will talk about the methods. So for predicting the dog breed, we use several machine learning classifiers. And for predicting the dog height and weight, we use several regressors. So as part of the classifiers, we also trained a multi-layer perceptron, both in linear and nonlinear modes. And as input to the models, we used different percentages of the dog DNA sequences, and which were randomly sampled. And we repeated this for 20 experiments. So these are the results of our study. So in the x-axis, we have different percentages of our input data with the exact number of SNPs that these percentages correspond to. And in the x-axis, we have the mean and standard deviation of the performance metrics. And each of the lines correspond to a different method. So we can see that for the breed classification task, all top 5 methods are nonlinear, and with only 0.02% of our data, so only with 40 SNPs, we already get a balanced accuracy of 50%. And using more than 0.5% of the SNPs doesn't seem to improve. And the same can be seen for the height and weight regression tasks. But in this case, nonlinear methods often match linear ones. In fact, the best model is the nearest neighbors for few SNPs, which is nonlinear. And we can see that with only 0.5% of our data, we already get a balanced accuracy of 0.8. So in conclusion, very few SNPs are necessary for predicting the breed and height and weight phenotypes. And since we saw that linear methods outperform nonlinear ones for the breed classification task, we can conclude that breed and SNPs have a linear relationship, whereas the same cannot be said for height and weight prediction. So in this case, it is not very clear, but probably height and weight and SNPs have a nonlinear relationship. So next I will talk about my second project, which consisted in designing a toolkit for merging genomic datasets. And first of all, I will start by defining the problem as a motivation. So we have two datasets which contain dog DNA sequences. The first dataset, we call it whole genome, and the second dataset, we call it array. And we can see that whole genome contains sequences that are very large, and array contains much shorter sequences. And these sequences, they have some common features, some common SNPs. I've marked them with the same texture, but there are some features that they are only in whole genome, or some features that they are only in array. So how can we merge both data sets? Because if we do it by the common features, most probably we will be losing a lot of information which might be relevant for future studies. So our proposed method is to use the common features, the common SNPs, to infer the missing SNPs. And this is possible in genomics because genomic data has correlations. So for this task, we use public software, which is called Beagle. So now we've got sequences that have the same length, but we cannot train any model with millions of features. And these sequences, they have millions of features. So we should subset some part of these sequences. And to do that, we subset to the same features as another company, Embark, which they performed an study in which they selected the features from whole genome that contained more variation and that were more important. So if we do this, now we've got sequences which are much shorter, of around 200k SNPs. And this is a summary of the pipeline I just explained with the imputation and subsetting models. And we can see two additional models that deal with efficiency and manipulating purposes. And here we have the merged dataset with all the samples from the green dataset and all the samples from the yellow dataset. So now we evaluate this merged dataset, and to do that, we perform a principal component analysis, and we plot the first two components. So here in the plot, each point corresponds to a sample, to a dog DNA sequence. So green points correspond to samples from the green dataset, and yellow points correspond to samples from the yellow dataset. And we can see that two clusters are formed. So there is a clear distinction between the samples that come from one data set and the other. And this shouldn't happen. So why does this happen? This happens because genomic data sets have ambiguities, they have inconsistencies, and they have errors. And these are due to mishaps in the lab during sequencing, or differences in the technologies used for sequencing. So we could improve this, but currently there is no tool available that allows merging genomic datasets without losing all non-matching features, and also while dealing with data quality. So we designed Merge Genome, which is a pipeline of pre-processing steps to merge two genomic datasets, and that includes two additional models, the clean model and the filter model. So the first model, in general terms, removes outlier samples, ambiguities and errors, and cleans inconsistencies. And the second model filters the features or SNPs that allow identifying the source of the data. So if a sample comes from the green dataset, or if a sample comes from the yellow dataset. And there shouldn't be any feature that allows solving this classification task between samples from one dataset and the other, because there shouldn't be any distinction between both datasets, and the distribution of zeros and ones in each of the SNPs should be similar. So in order to identify these features that allow solving this classification task, which are likely erroneous, we use L1 regularized logistic regression, and we use L1 regularization because this type of regularization forces a lot of parameters to be equal to 0. So basically what we do is we identify the coefficients of the parameters or of the features or SNPs that are higher, that are different from 0, and that have a higher mean, a higher absolute value. And once we identify them, we remove them, and we repeat this iteratively for 100 iterations. So next, I'm going to show you the evaluation of the merged dataset, and also the results of the filtering block. So this is the same PCA as before, but remember that before we were seeing two clusters, one here and one here, and there was a clear distinction between the samples that come from one dataset and the other. And we can see that now, with all the cleaning we did, now both datasets they intersect a lot, the samples, which suggests that the merging was correct. We also made a plot with a comparison of the SNP means. So this helps evaluating the quality of the merging, and also the quality of the imputation with Beagle. So if you remember, I said that the distribution of zeros and ones should be similar between both datasets. So the SNP means should also be similar. So all points should fall around this x equal y line. And we can see that this is the case for the vast majority of SNPs, which indicates that the merging was correct. But also we can see that there are some outliers here and here especially. And these outliers, they correspond to imputed SNPs with Beagle. And what is happening is that for the SNPs that in whole genome they had a mean lower than 0.5, Beagle is putting even more zeros than it should, so the mean is lower. And similar thing happens with SNPs that have a SNP mean higher than 0.5 in whole genome. So in this case, Beagle is putting more ones than it should, so that is why the mean is increased. So this is a shortcoming we identified from Beagle. So in order to try to solve this, we tried several multi-label machine learning classifiers for the task of genotype imputation, but none of the methods tried outperform Beagle, so we leave this as future work. And finally, I'm going to talk about the evaluation of the filter model. So we repeated 10 experiments for 10 different seeds. And here I'm showing the mean and standard deviation of the balanced accuracy. So we can see that before, since there were a lot of outliers and some features that they were incorrect, the balanced accuracy was of 1, so really high. But as more iterations go by, it is decreased and then it stabilizes, which is the result we expected. So the filtering block, it works pretty well. So in conclusion, we designed a new toolkit for merging genomic datasets that it also targets data quality and has the advantage of scaling to any organism. So it doesn't work only with dog DNA sequences, but it can also work with humans data. And we also designed a new system for filtering the SNPs that allow identifying the source of the dataset, which are likely erroneous. And this filtering model also serves as an evaluation of the quality of the merged dataset. And finally, we trained several supervised machine learning classifiers, but they did not outperform Beagle for the genotype imputation task. So we leave this as future work. And well, that would be all, but I would also like to add that we want to make the code public of merge genome so that anyone that wants to merge two datasets with genomic data can do it. And thank you for your attention. And if you have any questions.
Thank you, Míriam. Let's start with the questions. Javi, do you want to start?
Remember to stop the
Para de
recording.
grabar.
Sí.
Again.
Míriam, first of all, let me tell you that,